I'd like to welcome Dr. Chen. Thank you for agreeing to do your presentation today. Dr. Chen is a tenured professor of biostatistics and the founding director of the Center of Health and AI Synthesis of Evidence, better known as TASTE, at the University of Pennsylvania. He's an elected fellow of the American Statistical Society, International Statistical Institute, Society for Research in Synthesis Methodology, American College of Medical Informatics, and American Medical Informatics Association. He founded the Penn Computer Inference and Learning Lab at the University of Pennsylvania, which focuses on the clinical evidence generation and evidence synthesis of using clinical and real-world data. During pandemic, Dr. Chen is serving as Biostatistics Core Director for a national multi-center studies on post-acute sequelae of SARS-CoV-2 infection, involving more than 9 million pediatric patients across 40 health systems. Thank you, everyone, and please welcome Dr. Chen.
Yeah, thank you, Judy, for the nice introduction. Thank you, everyone, for being here. So I'm going to spend the next hour with you all to talk about this recent work that we have conducted regarding real-world effectiveness of BNT162b2, this COVID vaccine among children and adolescents. And we will cover some scientific component of this work as well as some methodological consideration. I want to begin with this disclosure. So I have the following financial relationship to disclose as serving as a consultant for multiple entities. So I want to begin with a very brief introduction about the real-world data and then talk about what is real-world evidence. So real-world data is data about the patient's health and care delivery is routinely collected by health systems. And there are lots of examples for real-world data: electronic health records, insurance claims, and some public health data and disease registry data. For today's talk, we are going to focus on EHR data. In particular, we will talk about some important limitations of EHR data and the strategies to account for this limitation when we conduct the inference using EHR data. So with EHR, with real-world data, we can extract real-world evidence. So those are the information derived from sources outside of typical clinical research settings, like clinical trials. And the goal is to generate reliable evidence from real-world data. So why real-world data and real-world evidence are important? In general, we can use this data after careful analysis and design analysis and inference. We can effectively evaluate the real-world effectiveness of medical products and as well as the safety of those medical products. So a signature study conducted based on real-world data, led by a group of Odyssey collaborators, is this Lancet paper published in 2019, led by Mark Suchard and colleagues. And in this Lancet paper, people conducted this study across multiple countries, and systematically, this study systematically evaluated as many as 6 effectiveness outcomes and 45 safety outcomes for all the first-line antihypertensive drugs. So not only this paper generated evidence in the sense of replicating what has been already done in clinical trial setting, it also generated new evidence for some comparisons that cannot be conducted in clinical trial context. So this is one example of using real-world data to generate reliable evidence. And another example is regarding the safety. So a great example is this Ralph Cox example. So back in 1999, FDA approved this drug for treating acute pain. So this drug is also known as Vioxx. So, and after this drug was approved, there was a widespread use of this medication. Over 80 million people are using this medication. And in US, there are more than 2 million patients being prescribed. And later on, there was a study called the VIGA study. Based on real-world data, they identified a significant increased risk in myocardial infarction. And this was a paper published at the New England Journal of Medicine. And it's reported that this medication, Vioxx, is associated with 4 to 5-fold increase in these myocardial infarctions. And after that, FDA provided this warning letter, required Merck to add this label about this potential risk, increased risk of heart-related complications. And then Merck conducted a study, a randomized control trial, and confirmed this evidence. And this drug was later on withdrawn from the market. So as you can see, real-world data can be really useful in terms of generating evidence regarding real-world effectiveness and safety, with a goal to provide additional real-world understanding of the efficacy and safety for clinical decision-making. So now to our, if we focus on these COVID-19 vaccines, and we know COVID-19 vaccine has been widely applied internationally and in the United States, and has a proven effect in terms of reduce the likelihood of being infected and prevent severe infection and decrease the hospitalization and the death rate. And on the other hand, if you look at the guideline across United States and other countries, there are actually quite different guidelines. And in United States, so CDC recommended the COVID vaccine for every single person more than 6 months old. And if you look at the guideline in Singapore, so it's quite different. We are talking the suggestion was for children aged 6 months to 4 years. I recommended 2 doses, and then children aged 5 years and to 17 years, and they recommend similarly. And then so there are some different suggestions in UK, and recommendations are for the children and the young people under 18 years at high risk for taking this vaccination. So as you can see, there are some slight, subtle, or some major differences regarding the recommendations. So the reason for these differences are multifold. There are local, the specific considerations across different countries, but also another reason is there are some inconsistent understanding about the effectiveness and the safety for COVID vaccines. So, so as a parent, typically the question we want to ask is, should my child get a COVID vaccine? And should our child get a second vaccine or a booster shot after the first one, right? Those are the practical questions. So if we now disentangle this question, basically there are 2 questions people we want to ask. The first question is, what is the real-world effectiveness or protection against the infection and the severe illness? And as we know, at the beginning of the pandemic, there were 2 major pediatric studies published at the New England Journal of Medicine. The first one was in 2021, and was focusing on the adolescents aged between 12 to 15 years old. And the second one was to focus on the younger children. And as we know from the early 2021, the pandemic, the variant also evolved over time. So that also added to the complexity. So what is relatively unknown is what is the real-world protection vaccine against the later variant, for example, Omicron, right? So rather than the Delta variant. And another major consideration is about safety, right? So there has been some reports about potential myocardial complications after the COVID vaccines. So there has been report from JAMA and also Internal Medicine, as well as the major journal Vaccine. So especially this has been a concern about, for the teen boys, that people are reporting some potential association with vaccination. So to tackle this question, we are actually in a unique position that from this large Recover consortia funded by NIH. So Recover consortia was launched about 3 years ago, and it covered as many as 40 health systems in the United States. And there are two major cohorts. One is this adult cohort, and led by N3C, and also the Weill Cornell, and the pediatric cohort covering as many as 10 million patients is led by the Children's Hospital of Philadelphia. And in this setting that we can access as many as 10 million pediatric data, is about more than 10% of the United States pediatric population. So we are really at a unique position to tackle this question scientifically using an evidence-based manner. So to do that, so we need to review this trial emulation pipeline, or the legend framework proposed by the Odyssey. So basically, there are 3 steps conceptually. First, we formulate carefully a clinical question. It could be an effectiveness study or safety study, or some other studies like heterogeneous treatment effects. And then you conceptualize an idea, a randomized control trial. And by filling all these key components, inclusion, exclusion criteria, treatment strategy, is specifically how would you define the treatment, what is the assignment, how long is the follow-up, and so on. And then using real-world data, we can try to emulate this randomized control trial by specific additional considerations. There are 2 parts that are particularly important. One is we need to emulate this blind assignment, where propensity score method has been proven very powerful. And the other is this the notion of so-called residual bias in real-world data, because the real-world data are truly messy. There could be some phenotyping error, the missing not at random situations. There are all kinds of potential biases in the real-world data. And so the negative control experiment is a set of tools developed by the Odyssey collaborators, where we kind of lump all this potential negative impact of from this real-world data, and then try to run this analysis regarding to control the potential residual bias that is due to the imperfect collection of the in the EHR data. So to give you an example of emulating a trial, so this was a paper published in 2022 at the New England Journal of Medicine. And this study used the VA data to evaluate the real-world effectiveness of Pfizer's vaccine and Moderna's vaccine. And basically, as I said earlier, we have these major protocol components, and then you can conceptualize a targeted trial by specifying the granular terms. And then the third column is to emulate those conceptualize trial by first following those specifications or providing some modifications or additional considerations. However, when we conduct this type of exercise for COVID-19 vaccine for children, there are some unique challenges. The challenge is the following. So the vaccine records in the EHR is actually quite fully measured. So as you can imagine, the children, adolescents, they can get vaccination at the hospital. They are primary care physicians and urgent care. But on the other hand, especially at the beginning of the pandemic, the kids can also get their vaccination from a pharmacy or a workplace, or even at the parking lot. They can potentially offer a vaccine. But those vaccination information was not sent to the EHR data, depending on the how this EHR data is linked in that particular state. In states like, for example, the Rhode Island or California, I know they have very good HRE health information exchange program where they can actually link their EHR data with HRE. Through this HRE program, they can have a better capture of the vaccination. But for some other states like Florida, so the linking their EHR data with some the vaccine registry database has been proven quite challenging. So so at the beginning of pandemic, we were facing this particular challenge where we don't have time to conduct this data linkage, and then so basically we need to conduct a reliable causal inference without this validation of the vaccination. So statistically, this is called a measurement error issue. So what is making this more complicated is actually the measurement error of the treatment. So let me explain to you what is the implication in a few slides. But this is a screenshot of the paper later published at the Annals of Internal Medicine. So led by my postdoc and student Qiong Wu and Jesse Tong. And this paper, after it got published, it actually got lots of media attention. And in this paper, we evaluated this real-world effectiveness among children and adolescents, and also their durability, and also as well as this association with potential cardiac complications. So so let's talk about this problem setting. So we have a problem of evaluating the association between vaccine, so the true treatment with the outcome. Here we can, the outcome can be documented in infection or severe infection or ICU visit. And the V star is a so-called error-prone capture of this vaccination. So it's a vaccination status from EHR data. And on the other hand, in EHR data, typically we have a high-dimensional confounders like demographic variables, healthcare utilization, and many clinical factors. So notice that here, before we have the capability of linking our EHR data with registry data, we actually do not have this true treatment status. So the observed data is only this blue part, excluding this gray column. And so if we have the true vaccination status, what would we do following this legend pipeline is first we calculate a high-dimensional propensity score model, and it's in Odyssey community, it's called a large-scale propensity score model. So and then we use one of the three strategies, stratification, matching, or weighting. And normally the first two are preferred from our earlier experience. And then the second step, we run this so-called outcome models. Based on, for example, we can run a stratified Poisson regression for the outcome Y, which is infection, and against this the vaccination status, as well as controlling for the propensity score strata. So that's the ideal situation if we know the true vaccination status. So now with this misclassification in vaccination, and if we blindly apply this legend pipeline, so as you can see here, we have a logistic regression model, propensity model actually for the surrogate of the vaccination status is V star. So, and then another implication is that when we run this outcome model, so this V star is basically a measurement error version of the true vaccination. So this will also introduce the bias, right? So that is, as you can see, the measurement error in vaccination status actually hits both steps in terms of the propensity score model in step 1 and the outcome model in step 2. So what is the consequence? You may be thinking about, how big this negative impact could be. So we actually run some simulation studies, and we let this sensitivity, which is a probability of the vaccine vaccine status in EHR data equal to 1, V star equal to 1, given the V equal to 1. So it's basically about the capturing of the true vaccine status. If this sensitivity vary from 70% to 95%, and if you know the true gold standard of the vaccine status running the earlier pipeline, and then you would get this vaccine effectiveness being around 80. And if you ignore that, so you will actually, you could get a bias as large as 10%. And as we know, the 10% in terms of vaccine effectiveness is large enough for some clinical decision-making or policy-making for stakeholders. So what we proposed when we tackle this problem is a parsimonious solution. At the beginning of the pandemic, our overall thinking is, let's try to develop a pipeline that ideally make a minimum parametric assumption, and it's as simple as possible to target a clinical journal. And later on, when we have more time, we can try to pursue more sophisticated statistical procedure to tackle this problem. So this pipeline is actually very simple. So we just divide and conquer. For the first step, as we know, the error-prone vaccine status is impacting the propensity score model. So if we can stratify the patient population, which is a case recommended by our pediatric collaborators, we stratify them by younger age between 5 to 12, and then adolescents between 12 to 21. And we found that the misclassification rate by some validation data is highly dependent on the age group. So if that is a primary stratification, if that is a primary impact of this misclassification rate, specifically this specificity, sensitivity, so we can assume this sensitivity to be a constant across different subpopulations. So this assumption may be approximately true. So that is an assumption we made. So basically, this diagram is showing the assumption. So as you can imagine, if someone's name is shown in the EHR database showing this guy got vaccinated, so we are quite certain this person has been vaccinated. On the other hand, if this person is not shown in the EHR database being vaccinated, so this person may or may not be vaccinated. So in this case, we have the specificity equal to 1, and then the sensitivity is less than 1. So it's basically an underreporting situation. So in other words, this misclassification in treatment is actually one-sided. And with this particular observation, so there's a very nice and simple mathematical property for propensity score model. So we can see if we are basically run a propensity score model, fit this model based on this error-prone vaccine status, and with a simple law of total probability argument and this non-differential misclassification assumption, so it's actually proportional to the true propensity score. So remember, earlier on, I talked about the 3 ways of using propensity score, right? So the stratification and the matching, as well as the weighting. So if we, in particular, pick this stratification, we have this very nice rank preservation property that if you, as you can see here, if you fit this, the propensity model based on the surrogate, this error-prone version of the vaccine, is going to be highly rank preserving compared to the true propensity score, which we do not observe. And the empirical version is visualized in the simulation on the right-hand side. And you can also look at this confusion table. If we are categorize the propensity score in 5 categories, you can see the majority of the patients are actually in this diagonal group, right? Diagonal part of this matrix. So that's the first part. So we are, we have a simple way to address the impact, the negative impact of the underreporting on propensity model if we can assume the non-differential misclassification. And the second step is this outcome model. So suppose we are using this stratified Poisson regression. So one way to tackle this is to use a technique called the integrated likelihood. If you are full Bayesian, you can think of this as a Bayesian model averaging. And the frequentist version of this is to basically integrate the likelihood function, which is used to make inference about the vaccine effectiveness through a range of models. And the definition in this particular case is that we can impose a model where theta is our treatment effect. So you specify a model regressing the outcome on the true vaccine status, where the coefficient is indexed by theta. And this gamma indexed this sensitivity. Basically, how good is this vaccine, the error-prone vaccination status compared to the true vaccination status. And this is considered as a nuisance parameter. And then you can integrate over informative prior that you can use. And in some earlier investigations that we have done before in a completely different setting about bias reduction, phenotyping error in EHR, we have showed that this can be a quite effective method to address the bias. So in our setting, so by working with a group of clinicians, we specified two different ranges of sensitivity for children and adolescents, and we use this as an informative prior to conduct this integrated likelihood inference. And we also conducted a sensitivity analysis by changing this range to a wider range and to study how sensitive is our results on this informative prior. So now with this new parsimonious prior knowledge guided CER pipeline, so we applied our method to this large distributed research network. And in this particular network of cohorts, we have about 6.2 million patients from 12 states. And so we decided to focus on the Pfizer vaccine because the Pfizer's vaccine actually covered more than 85% of vaccine records in the PEDSnet database. And we also conducted an analysis using all the vaccine, including those additional 15% as a sensitivity analysis. And so with all this setup, we have about 250,000 patients in the study cohort. So back to the two original questions we asked, should my child get the COVID vaccine? In terms of the effectiveness, we listed this, we studied both the Delta period and Omicron period. Since Omicron is more, more recent, we reported this Omicron in this talk. So the overall protection against the documented infection is about 74%. As well as this for mild COVID-19 and on moderate, we stratify this by severity, as well as the ICU admission. So the for children, the Pfizer's vaccine has a high level of protection around 85% against ICU admission. For adolescents, there is a higher degree of protection. So the numbers effective, the efficacy is about 85%. And this number is about 92% against the ICU admission for adolescents. So we also studied this durability because a practical question for parents like us is, should they, after they got the first vaccine, should they get a second one, right? What is the durability of this vaccine? So known as the waning effect, right? So we found that in the first 4 months, actually the Pfizer vaccine has quite a good protection against the Omicron among children. And after 4 months, there is about 10% drop in this protection against the infection. And we also studied this dose response relationship. For example, 2 doses versus a single dose. And there is a sizable benefit for having the second dose. Okay. And a more relevant question, for parents of teen boys in particular, is regarding this safety, especially against the cardiac complication. So we found that the relative risk for children and adolescents in terms of against this cardiac complication is actually quite substantial. There is about 72% of protection against those cardiac complication and about 90% of protection. On the other hand, you can see the confidence interval is quite wide because the cardiac complication is actually highly rare. Even with 6.2 million patients, we are talking about the events in the range of 5 to 15. So it's a really small number. This is a really small number of events happened in this population. So fortunately, and some possible explanation as we look into the literature, there has been some reports earlier, including our earlier report to CDC, this MMWR report. So the rationale is that the protection against this cardiac complication due to vaccine could be largely attributable to the protection against this COVID infection, because COVID infection is known to be associated with cardiac complications. So just to summarize the first part of the talk, so this paper, in this paper, we implemented a new prior knowledge guided comparative effectiveness research pipeline for bias reduction. And this is one of the largest study in pediatric population, and by far it has the longest follow-up, so that we can study the durability of the protection of vaccine for pediatric population. So now, if we are moving forward, think about when we get to this later part of the pandemic, when we have more resource and manpower to actually link the data. So link the EHR data with the registry data. So there's an opportunity that we can do better. So this is the simulation results. So regarding the sensitivity of our analysis with respect to different prior knowledge, different level of prior knowledge. So the gold standard, again, the gold standard is in this red dots. So for you can see that it's around 80% effectiveness or efficacy. And if you completely ignore the underreporting issue for vaccine, so you will get quite a biased estimate of the true vaccination, vaccine effectiveness. And now using different types of the prior knowledge, so we can have quite a substantial bias reduction, but still there are some gaps. So the question is, can we do better if we have validation data? So quickly recap what we have done before. So when we don't have any linked data, so this framework is easy to implement and good for some time-sensitive research when we do not have the time or the resource to link the data with validation data. And we are focusing on bias reduction, not bias correction. And a key assumption made here, which is a limitation, is the non-differential misclassification. So we have to run lots of sensitivity analysis to see what if there are some differential misclassification, how much is the result will vary. Of course, how much is the result vary depends on the type of model we specify and the space of the parameter we explore. And now if we have the data linkage, and we actually have some registered data for some of our sites. For example, our clinical site in Colorado and clinical site in California, they actually have those validation data set. In other words, a part of the patients have their true vaccination status. And then the goal is to use this contrast. So we start to calibrate for the analysis for the entire population. So one naive solution is that we can just pick those hospitals with a gold standard, with the linked data. By this way, we will throw away lots of patients from the data, and also this analysis may not be the coverage of this population may not be representative or inclusive enough. So we want to calibrate by using the contrast between V and V star. Okay. So this is the ongoing work and being a bit more methodological. So the idea is, as I said, using this internal validation. So we can formulate this problem using some standard causal notation. So here, this R is a selection of internal validation. So it's basically a missing data indicator. So R equal to 1 for those patients, and R equal to 0 for the patients that cannot have their linked data. And then we can denote using this notation to denote all the observed data, again, using Y1 and Y0 to denote the potential outcome. In this case, it can be documented infection or severe infection or ICU visit. And again, our estimate is this average treatment effect, the ATE. So ATE is defined by the difference or contrast between these two potential outcomes in the population level. And the typical assumption made in the causal inference literature is the consistency, the ignorability, and the positivity. And there are some additional assumptions we made here. So we need to assume the vaccination status captured by EHR data is non-differential to potential outcome given the covariates and the true vaccination status. And we also need to have this random selection validation. For now, we just assume the part of the patients who have the validation data, like the California and Colorado, they are random among the different state of populations. And as we know, this actually has largely to do with political leaning, Republican versus Democratic states, right? So, and also we need to have this positivity of the selection. And we found that in the econometrics literature, there was a really elegant paper written by Arthur Lubell from Boston College. So basically in this paper, the main results he established, one of the main results is the non-parametric identification. So in other words, if you know this sensitivity model, or the PPV, actually, this is a PPV and this is 1 minus NPV for the true vaccination status. If you know this, how this depends on X, so you can identify the average treatment effect with this term. And later in his paper, he used instrumental variable to conduct the inference. And so in our context, so we are in a better situation that we have partial validated data. So we can actually use that partial validated data to calibrate and use that identification result to establish a semi-parametric efficient estimator for the average treatment effect. And what is important in our analysis is that this semi-parametric efficient estimator is actually convergent in the rate of the total sample size, meaning that we are talking about the error rate using the entire population rather than only the internal validation population. Okay, so just to summarize what we have done, so the finished work, which is in this Annals of Internal Medicine paper, we provide a bias reduction approach when we don't have any linked data. And it's easy to implement, but it suffers from the limitation of its focus on bias reduction, not correction, and also it relies on this assumption of non-differential misclassification. And whenever we have the ability to do this data linkage, which is always recommended, and we can leverage the partial linked data to conduct this more sophisticated bias reduction, where we don't need to make a non-differential misclassification assumption, and we can achieve the full statistical efficiency. Okay, and just to conclude this talk, so in general, this underreporting of the vaccine status, can lead to severe bias in estimated effectiveness and safety. And so this type of challenge may not be unique associated with vaccine. So it can also happen in the misclassification in treatment. In some other situations, either the treatment or the dosage information in EHR may be questionable, right? So that's again a typical measurement error plus the causal inference problem. And we are interested in extending this framework to tackle those additional scenarios. And so without validation data, we do have a pipeline and ready to implement. And with validation data, we are currently developing this bias reduction correction tool, and ongoing work. This is an ongoing work in terms of developing this software and and running this analysis to this large linked EHR data. So with that, I'm going to stop, and I'm ready to answer any questions you may have. So thank you.
Thank you, Dr. Chen, for that wonderful talk. We do have a few questions. I'm going to start with ones here in our chat. Kevin, if you're still on.
Oh, sure. Yong Chen, that was a great presentation, great overview. I was wondering about how this impacts with outcome misclassification. So you in theory also have a Y star where you have many outcomes, particularly mild COVID or even the myocarditis, which would happen well outside of the PEDSnet realm of knowledge, right? And so you have that piece. And then you also have the positive predictive value of an actual outcome. So just because you saw the outcome, then was it valid? Can you talk a little bit about the non-differential and differential impacts of outcome misclassification, and especially as you see over time, people then may seek care outside of the healthcare system. So I got my kids vaccinated at CVS, but I made sure that they had their COVID outcomes at Nemours. We're CHOP kids, but then we had our myocarditis at Nationwide, just to completely mess with your data, right? The fragmentation of the US healthcare system has equal problems on the outcome. So your Y and Y star, and then if you see a Y, is that Y real?
Right. That's an excellent question. Thank you, Kevin. This is like the scenario talk about, there are two layers. I think there's a first layer of outcome misclassification. And the second layer is, we call it in informatics community, the data fragmentation, right? So meaning that our database only capture the part of the patient's episodes, right? So the data fragmentation is a much harder problem. If you only capture a part of the patient episodes. So, for example, the loss of follow-up, we statistically we could consider this as a censoring, or but we should also be very mindful when we define the inclusion exclusion criteria. Make sure we have a unbiased capture of the patient cohort that can be used to study the real-world effectiveness. So that's, I think, to my knowledge, that's the best we can do to handle this data fragmentation. And luckily, this PEDSnet has a very relatively good catchment, for the patient population. Again, we cannot exclude. It's not like a VA or Kaiser Permanente. It's more like a closed system, right? It's very good, So VA would be suffer less about this fragmentation issue. And back to your first question about the outcome misclassification, that was actually a question asked more than one time from the reviewers. And so people's behavior are different. At the beginning of pandemic, people, whenever I got a pandemic early on, so I got this the COVID actually in the kind of a peak season of the pandemic. I reported to my my PCP, but later phase, they don't care, right? People just say, okay, it just let the kids to quarantine themselves. They don't report. So there is a varying degree of the capturing the quality of the capture of the outcome in the early phase compared to the later phase. I think that one thing we could do is to run some sensitivity analysis in the by cutting the data by different quarters of the year, calendar year, and to see how results how much they matter. But we do not have, in general, we do not have the some type of gold standard for the outcome. So it's very hard to validate in this database.
Well, I mean, one of the strengths would be you linked in 2 states to the IISs, I imagine, to get that better capture of the vaccine status. The alternative would also be to link to administrative claims to have that longitudinal capture of care fragmented outside of the healthcare delivery system and see the impact of being able to identify events that were outside of your purview.
Right. That's a great idea. So we will have some partial validation, right, for the both outcome and the vaccine status, and likely that can be done in the similar hospital, right? So we will find out. That's a great question. Thank you, Kevin.
Thank you for that, Kevin. So we have another question from Mark Suchard. It says, it appears that both approaches assumes non-differential misclassification. What do you see? What hopes do you see for advances in quantifying and then modeling differential effects?
And in particular, Yang, I'm very excited about the Bayesian models that you're doing. But you have, you kind of fixed specificity as one, and that makes it a simpler problem. What ideas do you have if we have kind of the bivariate or the 4-variate for specificity, sensitivity, and those might vary for both groups? I mean, what type of Bayesian models might you be looking at?
Right. First of all, yeah, great questions. So first of all, I need to clarify the second method can handle differential misclassification. We do not make assumption about, for example, sensitivity is a constant across different populations. So just for clarification, the first part, back to the Bayesian model or integrated likelihood version, that's the frequentist version of this Bayesian model, is that we choose to use the unified model for model parsimony and simplicity. And you could imagine situation that the specificity may not be exactly at 1. So maybe we should allow some range of parameters for specificity being close to 1, but not to exactly to be 1. And I have not explored the Bayesian implementation. So right now, since it's one-dimensional, it's actually pretty easy to implement when you have multiple parameters involved in this integrated likelihood. So that actually gets to the point that we may just want to run a full Bayesian method rather than a integrated likelihood method. But previously, in a different setting, I tried a multi-dimensional integrated likelihood. The implementation is actually quite subtle. A technical comment is that you actually integrate the likelihood function over a range of the possible value for parameter, rather than a log likelihood function. So as you know, when sample size get large, this likelihood function will be very, very tiny. It will be very spiky. So there are some work done by Severinis from Northwestern and his student. They use some Bayesian ABC method and some other method, but that's getting kind of beyond my knowledge of Bayesian inference. So I'd love to discuss with you more about this. So,
thank you very much, Yang.
Thank you.
So another question in the chat. This is regarding going back to slide 22. There's some useful information there.
Right. Let me share
like
this.
to unmute to ask your question. That's perfectly fine with me. I can go ahead and unmute you, Hugh.
So slide 22.
So
what was the question? Slides. The property. Yes, go ahead.
No, that was just George repeating what's in the chat. It's just saying it's a very useful property, presumably the rank preserving property, and maybe saying a little more about it.
Right. Yeah, thank you, George. So this is actually quite a simple result, and you can derive this by so-called law of total probability by adding V to this probability calculation. And but with some a few lines of calculation, you can see this propensity score for that you build with respect to using the error-prone vaccine status is actually related to the true propensity score for the same patient up to a constant. And this relation is also true when specificity is less than 1. So then the only thing you would have is an intercept. So in other words, it's still a linear function in the sense that it will preserve the ranking of the propensity score. As long as the non-differential misclassification is true, we always have this nice property. So this is I don't know whether this has been reported in the traditional causal inference literature, but this indeed is a cute, nice, simple result we found by this simple exercise. And we are basically trying to make use of this property. And then because originally the goal is to stratify based on the true propensity score, since we do not observe this true propensity score in this green box, and since we have this rank preserving property, we can just stratify by the propensity score derived in this red box. So that's the whole idea. And in the simulation where we have both, we have a true vaccine and the error-prone vaccine, we can cross-tabulate them. And you can see there's a high concordance between these two values. So we feel quite confident by working with the propensity score built by error-prone vaccine status. So that's the main finding here. So
Yong, may I ask, and thank you for the wonderful presentation and thank you for the wonderful paper. Given this property is, generically useful for us to know about, in other words, if we do PS stratification, in our, say, drug effectiveness studies, at what point do we want to use more of your techniques in drug effectiveness? Or do you think that the misclassification is low enough that this is really for vaccines where we're really getting it wrong a significant portion of the time?
Right. So I think, again, the key assumption is relying on this non-differential misclassification, right? It depends on how much we believe this assumption can be true, and we can also run a set of simulation study to explore to a certain degree some differential misclassification. In this particular case, there was actually a paper published by our colleague Hania at CHOP. She actually contrasted this error-prone vaccination versus this some linked data, and she found out that the main modification for sensitivity is actually the age group. So that's why we feel that might be reasonable, since we are going to stratify the age group for clinical reason, biological reason, because they think vaccine impacted the children, protected the different ages of children differently. So that's a nice coincidence. And back to the treatment evaluation, again, it's highly dependent on the non-differential misclassification assumption. I wish if there is for example, some partial validation, if we do have partial validation, let me go to my one slide. So seems that there are two things we can do. So with partial validation, we can go through this semi-parametric exercise to do the derivation. And on the other hand, if we using this contrast, we can fit V. We can fit V star given V and X and study the sensitivity. How is the sensitivity dependent on X? If at the end of the day, for this small data set, we find that the coefficient for X are close to 0, that could give us some confidence of using the more parsimonious method, right? So, but I would generally recommend doing some diagnosis using some type of partial validated data.
Hey, Jan, this is Thomas. I have a quick question. Do you mean when you say non-differential misclassification, is that just meaning that the error rate has a uniform distribution throughout the population?
Right.
Okay. Okay.
Yes. Thank you.
And yeah, Jody, we just talk, right? Or do we have to first type it in? Yeah. Yeah, I agree. It's very nice. Very nice work. The partial validation using the registry, as opposed to, do a chart reviews or anything like this. I actually don't know how these registries work. Do you only have vaccinated patients in there? Or do you have some patients and you know that they're vaccinated or not vaccinated? So is it also only one-sided? Or is it, do you also know the cases which you are missing? You know what I'm saying?
Yes, I know. So I had some extended discussion with, in particular, with one of the smallest states in United States, Rhode Island. They actually have a very nice integrated database, and Elizabeth Chen and Neil Saka, they seems that in Rhode Island they actually have their EHR data directly linked with some registry data. Whenever you have some child being vaccinated, they will send that information to their EHR system. They will overwrite the vaccination status as a new data attribute. And regarding the more detailed operation, like the workflow, that's beyond my knowledge. So. No more questions.
Young, did you happen to look at effectiveness at 1 week? I'm just curious because some say it should be no effectiveness at 1 week, whereas we found that it should be artificially high at 1 week.
Right. We did
found.
not. No, we did not. We only look at for the outcome. We purposely defined it had to be anything happen after 2 weeks.
Okay.
Yeah.
Thank you.
Thank you. It might be interesting to look into that. So.
I'm scanning our chat here for additional questions. I don't see any. I'm just going to give folks an additional minute before we conclude. Feel free to raise your hand or add your questions into the Q&A option if you're too shy to unmute. I see no takers. So with that, I would like to thank Yong for presenting today. And please join us again for our next installment of the CBER Seminar Series. Thank you for your wonderful presentation.
Yeah, thank you for being here. Bye-bye.
Bye, everyone.